This video shows the process of the embryo biopsy procedure. And in the first step of this procedure, we are preparing a dish which is used to biopsy the embryos. We have two main components of this dish making, the first of which is the culture media or the buffered media that the embryos are placed in during the biopsy. And the second one is the oil overlay. So as you can see right now, the embryologist is individually placing each drop onto the dish with a sterile pipette. And then in the second part of this process, you will see the embryologist use a second pipette to add this oil overlay. And this entire embryo process is performed at 37 degrees, which is comparable to body temperature. And this oil overlay helps ensure that the temperature remains at 37 degrees and that the drops themselves do not evaporate. Once the dish preparation is complete, the media is returned, and the second step is the QC of the laser system. And you can see here a continual line of ablation occurring upon the screen. And that is just to verify the alignment of the laser to make sure that the actual ablation lines up with that red dot. And that is to ensure precision when we are biopsying the embryo. And all of this is occurring at about 400 times magnification and all performed through the objectives of the microscope. The next step is adding your micropipettes to the microscope. And there are two of these. The first one is the holding pipette, which is approximately 120 microns. The second one is the embryo biopsy pipette, which is about 26 microns. The holding pipette is used to secure the embryo from moving during biopsy, and the biopsy pipette is used to remove the cells themselves. So now the embryologist has gone to the incubator to collect the culture dish with the embryos. A second embryologist is called over to verify the patient information is correct. And now the embryologist will move the embryos for biopsy from the culture dish to the embryo biopsy dish that you saw prepared. And any time before an embryos are moved, the device itself is checked to make sure it's working and operating appropriately. Once this is verified, we then use this device to move our embryos. Now you can see the embryo being placed into the biopsy dish in a very specific drop. The culture dish is then returned to the incubator with the remaining embryos that are not being biopsied. Once the embryologist adds the dish to the microscope, you can now see that the pipette tips are over the dish, and we use three-dimensional joysticks and motorized movements to control these pipettes. There is also fluid movement within the pipettes themselves to help secure the embryo and control the cells. The embryologist is verifying the alignment. You want everything to be precise. The next step will be for the embryo to be attached to the holding pipette, and the embryologist picks a very specific location to biopsy the embryo. So now what you see is to the left of the screen is the holding pipette and the embryo, and what you see on the screen is the hatching part of the embryo. Now you can see the biopsy pipette has come to connect with the embryo. The goal is to get about 6 to 8 cells within that biopsy tip. Once that has occurred, the embryologist will use the laser system to ablate the cells and remove them from the embryo itself. This usually occurs with a few pulses in a vertical line. And you can see slowly the cells removing themselves from the embryo. And now in this screen, you can see that they are two separate entities, the 6 to 8 cells, and then the embryo itself, which is composed of about 200 cells. And the embryo still looks healthy and happy. And the next step is to remove that embryo and place it back into the incubator. So you can see the embryologist grabbing a post-biopsy dish. And this dish will be labeled with the patient information and the embryo number. There will also be a second witness to verify this information is correct. And the embryo that was just biopsied will now be placed into this dish and then back into the incubator, and it will await the freezing process, also known as vitrification. The cells that were biopsied remain in this biopsy dish. As you can see, the embryologist moving that to the top left. In this last step, you see that we now have our genetic testing kit, which is that green box. And this kit is what is sent off to the genetic processing laboratory. This kit is labeled with patient information and also the date of the biopsy itself. And this is all done under a sterile hood using sterile gloves, and a mask is also worn to create a safe environment. After the kit itself is labeled, the next step is then to label the tube. Labeling the tube consists of embryo number and the patient's initials. Now the embryologist is getting their device ready, which they will use to move the cells from the biopsy dish to the tube. And before it is used, it's also verified that it is operating and functioning appropriately. The embryologist now has the buffer that is used to rinse these cells and is what they are sent in the biopsy tube. You can see the embryologist using a sterile pipette to create drops, and each set of cells will go through a series of these drops. In the left hand of the embryologist, you will now see the PCR tube. And next you will see the embryologist adding the buffer with the cells into that PCR tube. This tube is now capped and placed into the kit, and the kit is securely closed and then placed into a freezer. With the cells frozen, they now wait to be picked up by the genetic testing company. And the last step is to complete the paperwork, make sure the genetic testing company has all the information they need, and our culture paperwork is updated as well.